All right, so it's 12:01 p.m. here in Melbourne, so let's get started. I first like to acknowledge the traditional owners of the land on which we are meeting, and I pay my respects to the elders, past and present, and the Aboriginal elders of other communities who may be here today. Good afternoon, everyone. Thank you for joining today's webinar titled Airborne Microplastics. I'm Sabrina Idris, a Centre for Safe Air Postdoctoral Research Fellow, and I'll be chairing today's webinar. Microplastics are now pervasive across nearly all environments, including the air we breathe. While the health and ecological impacts of microplastics in water and in food have received significant attention, airborne microplastics is an emerging area of research with growing relevance for health. The respiratory tract is a primary interface with the environment, with the average person inhaling around 11,000 liters of air each day. As such, airborne micro and nanoplastics have the potential for extensive health implications. In this webinar, we will discuss new findings regarding the extent and health implications of airborne microplastic exposure, with presentations from distinguished Professor Lidia Morawska and Dr. Fatma Noor Eraslan. Each speaker will present for up to 20 minutes, followed by a Q&A after all presentations have ended, so please drop your questions in the Q&A section. Our first speaker is Lidia Morawska, who is a distinguished professor at the Queensland University of Technology, QUT. She is the chief investigator of the Centre for Safe Air, director of the International Laboratory for Air Quality and Health at QUT, which is a collaborating centre of the WHO on research and training in the field of air quality and health, and the Centre Director for the ARC Training Centre for Advanced Building Systems Against Airborne Infection Transmission, also known as THRIVE. She conducts fundamental and applied research in the interdisciplinary field of air quality and its impact on human health and the environment, with a specific focus on science of airborne particulate matter. She is an author of over 1,200 journal papers, book chapters, and refereed conference papers, and has been involved at the executive level with a number of professional bodies, including acting as an advisor to the World Health Organization. Over to you, Lydia. Would you like to share your screen?
Many thanks, Sabrina, and thanks for inviting me to give the seminar. And first of all, in a way, a big disclaimer. I'm not really an expert in microplastics or in plastics for that matter. I have never been really actively looking for involvement in this field, but somehow this has been coming my way, causing each time extreme confusion and frustration, dependently from each angle I looked at this. So I'll say first a few words about plastics in the environment, the source of microplastics. Then I'll talk about a little bit more about airborne particles and why size matters, because that's why we are talking about micro and nanoplastics. WHO report on nano and microplastics, a case study or studies in which I was involved in China, and finally a review which we've just finished, and the paper has been just accepted a day or 2 days ago, on the mechanism of plastic fragmentation. So the first time I sort of really started thinking about this topic was in 2019, Solomon Islands, this idyllic beach. We were there to investigate household air pollution, and our host took us to a remote community to discuss this project. And then I looked at this beach, and that's how it looked as far as I could see in all directions. I asked my host, where is this coming from? Well, they said part of this coming from Honiara, the capital, quite far away, because there's no garbage waste collection. But they thought a lot is coming by currents, ocean currents. This was really a shock, because this beach was so beautiful and so kind of idyllic. I'll come back to my research, but what I've been reading and considering in this topic is what's happening in general scale. An extremely frustrating development took place in August last year, when the treaty reached a deadlock on plastics and collapsed. Now, this is a article in the conversation about the reasons for this why it happened. But again, basically the main reasons is the fossil fuel production nations, the industry vetoed measures, including capping plastic production. So this is basically industry interest in plastic production, particularly fossil fuel in this industry, as the world is moving towards clean energies, less fossil fuels than fossil fuels towards production. This was a huge, really blow to the world. If you look what's been happening, again, it's mind-boggling. This is an article from the Scientific American, very recent, showing plastic production over time. So first invented in 1907, and then the increase of plastic production between 1950 and 2020 was from 2 to 473 million tons. And that other statistics, more than 10 billion tons of plastics were produced, and its acceleration of this. And also comparing this amount, what does it mean? Because we don't think about such huge numbers, twice the combined mass of all living land and marine animals. There may be a perception that plastics can be recycled, and that's what the industry is trying to tell us. But that's not the case. Very small fraction of plastics can, in fact, be recycled. Not that it is recycled, but it can be recycled. So reducing of use of plastic is an almost urgent matter, and it can be done at national level. We don't have to wait for international treaties of this. So that's a message for all of us. So airborne particles, since we are talking about the stuff in the air, the complex world of airborne particles, and however we define them, PM2.5, PM10, ultrafine particles, and whatever those particles are, whether it's black carbons, secondary organic aerosols, metals, desert dust, pathogen, and finally plastics, microplastics, again, however we'll define them. So when we try to collect stuff in the air, plastics in the air, we are facing this mix which is in the air, and particles, plastic particles, are only some of this. We can look at the sizes in many different ways, comparing the sizes of the smallest particles from combustion emissions like vehicle emissions through viruses, dust, bacteria, and then there are what's called microplastics. I come back to this definition in a moment in the context of these sizes. Now, this is something which I show in my lectures, comparing a sand grain with a boulder. If you were asked to measure a sand grain and a boulder, you would know very well that you must use different techniques. But if you measure particles in the air, many don't think that you need to use different techniques. But the difference in size between petrol exhaust particle is dust is orders of magnitude, like between sand grain and a boulder. You must use vastly different techniques to characterize these particles. Now, then there's an issue of terminology, which may sound boring, but then you'll see the terminology in relation to microplastics. So aerosol is solid and liquid particles suspended in gaseous medium long enough and so on. But the general issue, the issue is that these particles, the aerosol particles, are in the size from 0.001, which is basically nanometers, to 100 micrometers. Why? Because any bigger stuff is basically not stable in the air, gravity takes it down. And there are other types of particles, so it is dust, these are bigger particles from mechanical breakage. Nanoplastics could be as well. Fogs and mist, liquid particles, again, different processes act on them. So the classification of particle sizes as we use them is such: total suspended particles, smaller than 100 micrometers, suspended. PM10, PM2.5, coarse particles, ultrafine particles, differently measured. Now, the point is that really, from the point of health, we look at what's down below PM10, not total suspended particles. And we'll come to this in a moment, why. But also the definitions of nano and ultrafine particles, since we are talking about nanoplastics. So these are ISO definitions. A nanoparticle is a particle with a nominal diameter smaller than 100 nanometers. But something very different, an ultrafine particle is a particle size about 100 nanometers diameter and less. You can see the difference. Everybody is looking at this. Of course, there's no difference in what the definitions say. It is in different fields they are used. Nanoparticle is used in material engineering, ultrafine particles in atmospheric sciences. But this just shows that issue of definitions. So, but really, since we are looking at this from the point of health, so it is deposition in the respiratory tract. And here we are talking about inhalable respiratory particles and thoracic. So the stuff we can inhale must be smaller than 10 micrometers. If it's bigger, it doesn't pass through the nose. So that's why quite some time ago, TSP was abandoned as a standard in relation to health protection, because which initially was because these particles are too big to be inhaled. So then we know we have this kind of curves showing the deposition in the respiratory tract, so mass median aerodynamic diameter deposition. And you can see that the smaller the particles, the deeper in the respiratory tract they are deposited. And then there's the 10 micrometer. So guiding health protection are WHO health guidelines, with the most recent edition update in 2021. I was privileged to be a co-chair of this activity, and the guidelines set new levels, and here we are seeing new levels much lower for PM2.5 and PM10. Now, based on what this was done? Based on exposure response or relationship from epidemiological studies. So these are some examples of epidemiological studies with the impacts on acute lower respiratory infections, mortality, and so on. So you can see that this was that second study, 41 different cohort studies that was ordered for preparation of the WHO guidelines, this data review. So we know that these are the impacts, and we know that this is to this specific size, particles smaller than PM2.5. So size matters, as we know. So plastic particles, nanoplastic, microplastics, however we call them, must be smaller than 10 micrometer to pass through our nose. Well, this brings me to this WHO report on NMP, nano and microplastic particles. Again, I didn't specifically put up my hand to be part of this. I was invited to join this. And when the activity was already somewhat advanced and they realized that they needed somebody who understands stuff in the air. So this is the cover page of this document published in 2022. Now let's have a look on the definitions on which I couldn't have much impact really. So what is microplastic? A material consisting of polymer, but dimensions from 1 nanometer to 5 millimeters. And the length ratio up to 15 millimeters. Now, what does it tell you in the context of what I just said? What it gets into through our nose. Well, we are talking about the boulders here, 5 millimeters. So what's called, even in this document, as a microplastic, it is not what we understand as something which can be inhaled, even so that's what we refer to. And nanoplastics are also much, much bigger than to what is defined otherwise, like the definitions I've showed you a moment ago in relation to health. So recommendations by this report, just several of them, were that the evidence is low, substantial limitations. Standard methods should be developed to improve the quality. There are no standard methods, and specifically of particles smaller than 10 micrometers, yes, and better understanding of toxicological effects of environmentally relevant nano and microplastics. So basically, that's what the report is saying. We know very, very little. So the next time when I was involved in something again, I was invited in that sense that a student from China expressed interest in conducting part of the study after collecting samples in China to do the analysis under my supervision in Brisbane. So there were two papers published then, I think one more later. And this was looking on what was called microplastics again in Beijing and Shanghai coastal and inland environment. So that's where it was. The way it was done in that first study, it was a relatively small number of samples collected, and I will query this whether that was enough. I think it was 20 something. And that's how these particles, microplastic particles, look. So it's not about their shape and so on, but look at the size, looked at the electron microscopy images. They are huge. They are really boulders. And most of them are, again, bigger than what can get into the nose. So from these samples, we were able to look at the number concentration of different specific compounds. And you can see, well, there are differences in concentrations between Beijing and Shanghai. In general, there are more in Beijing than in Shanghai. In just of 2 or 3 of them, there's a little bit more in Shanghai. Now, in terms of average size, that's what the study found. So this is the sizes, so the types of microplastics and particle size in micrometers. So you can see that average size is well above what we can inhale. But again, we could not say that that's how it is. This is because this was the technique used for this, which is electron microscopy. So I just clicked on this again. Now, so there was a then second study looking at sampling sites within mega cities in northern China. So these are the cities. And the study looked in general about population, GDP, industry, and so on, which I'm not going to talk about here. But again, the size of different types found through the same type of sampling. And again, as you can see, this average size is much bigger than what can be inhaled. Different than microplastic in different size ranges. So we could spend a lot of time on discussing the reasons for this, and there's more explanation in the paper. But again, if you look at the size, these particles are big. They are in the air. They can be sampled in the air, but they are big. So this brings me to the last topic, which is an insight into physical fragmentation of plastics into airborne microplastics. Now, this was a review, which, as I said, was just accepted for publication. And we did this review because it was kind of, I wouldn't say pressure, but request. Why Thrive is not considering or habits in program something about microplastics in air, in indoor air. So I said, well, let's look in this. Can we do this? What's known? So so we specifically wanted to look about this current state of knowledge of mechanism responsible for particles getting small. Because if these particles are not below 10 micrometers, well, then they cannot be inhaled. So this was another jungle of well over 200 studies reviewed and finding out all kinds of things very difficult to compare. So first of all, we identified specific mechanisms which we were considering. I later mentioned what we didn't consider. We looked at laboratory settings and real life settings in general. If anything was quantitatively properly measured, it was laboratory, very little in real life settings. So then we looked specifically at what was involved in this. Not really quantitatively, because this aspect is not quantitative, but just understanding the mechanisms. So there are several images of this. So in particular, this deterioration of indoor flooring, microplastics to release materials, release microplastics. So that's what we expect if we walk and the friction between shoe and the floor. So that's one expected and investigated mechanism of fragmentation. Or release of microplastics from synthetic textiles due to well, wear and tear. So this shows steps in this. So peeling formation through the fabric, then fiber fatigue and abrasion, broken fibers, plastic release. So that's another important mechanism occurring all the time, every moment. Now, as we are sitting, moving our hands in fabrics which we wear, there is some plastic release. Fragmentation to mechanisms of tire. So again, this is the tire on the road and tire surface and how this process takes place. Again, it is kind of expected, but that's the mechanism is presented here. Part of this is actually an interesting aspect. It is not just mechanical removing these particles from the road, but through the temperature, between high temperature, particularly during aggressive braking, release of volatile organic compounds, which then form these secondary particles, microplastics. Or the release of inhalable microplastics from dried polymetric paint coatings. So again, the deterioration of this, how it gets into the air. I think these are all graphs we have. We have quite several tables. Oh no, sorry, there's one more, the formation of growth of plastic from 3D printing. Some years ago, we did investigations on release from printing, from any kind of printing, not just 3D printing, due to this process, high temperature, emission of volatile organic compounds, and then condensation and formation of particles. So then we put this and we tried this kind of something to quantitatively characterize. And this was extremely difficult because every study did something differently, analyzed things differently, and presented, if they presented concentrations, that concentration was presented in a different way. I'm not going to talk about individuals of this, but it was basically impossible to compare any of these studies. So the overall conclusion, that's the frustration. So the overall conclusion from this review is the fragmentation mechanism of plastic in airborne microplastics is influenced by material characteristics, environmental factors, and human or biological activities. Even more difficult to study are environmental factors such as UV radiation, temperature fluctuations, humidity change, and so on, environmental factors. So what we didn't cover, and that's not included, is the formation pathways and atmospheric behavior of environmental this was called nanoplastics. That was in relation to this text, and again the definition. It's another issue. So that was basically impossible, which means basically smaller particles. This was basically impossible because there's very little published in this. So conclusion number 2, most studies reported the fragmentation process, including all these processes I just mentioned. They reported that this size of particles, more than 10 micrometers, were generated. However, most were laboratory studies, and the conditions that we look at this did not reflect at all real world conditions and emissions. Most studies do not report the concentration of inhalable microplastics, which in this case we are talking about the 110 micrometers. So the current state of knowledge is very, very limited. And the final conclusion, microplastics are present in all environments, even the most remote, due to the global dispersion, and overall a very complex picture regarding the characterization of the microplastic in indoor and outdoor environment. Looking and understanding of this discouraged me to start a program within Thrive to tackle this. It's kind of a too big task. So to finish, the question is, airborne plastic particles are everywhere, but are they small enough to pass through the nose? Thank you very much.
Thank you so much, Lydia, for sharing an excellent overview of airborne microplastics. I also like to highlight that Lydia co-authored a Senate committee submission on microplastics, which Bill will share the link in the chat. There are some questions also in the chat, but we'll get to them after all the presentations have ended. Our next speaker is Dr. Fatma Nur Raslan, who is a postdoctoral research fellow at the Queensland Alliance for Environmental Health Sciences at the University of Queensland. Her PhD research focused on developing a green and efficient oleophilic sampling method for airborne microplastics in indoor and outdoor environments. Her current research focuses on developing analytical methods for the detection and quantification of micro and nanoplastics in human tissues. Over to you, Fatma.
Thank you so much to introduce me. Thank you so much. Thank you for everyone to be here today. First of all, I would like to acknowledge the traditional owners of the lands on which I am presenting today and on which part of this research was conducted. And I pay my respects to their elders and past and present. Today, I would like to share with you a new sampling approach that we developed to address one of the key challenges in airborne microplastic research. So, first, let's briefly look at what microplastics are. Lydia has already mentioned that, but I will just briefly talk about this topic. Microplastics are simply very small plastic particles, usually smaller than 5 millimeters, as we know. They can come from two main sources. Some are produced directly in small sizes, for example, in cosmetics or industrial applications. These are called primary microplastics. The others are formed when larger plastic items break down over time due to sunlight, weathering, or physical processes. These are known as secondary microplastics. Because microplastics are so widely used in everyday life, microplastics are now found almost everywhere in the environment, and more recently they have also been detected in the atmosphere. So why does this matter? This presence of microplastics in air is important for several reasons. First, the atmosphere can act as a transport pathway, meaning these particles can travel long distances from their original sources. Second, airborne microplastics may contribute to human exposure through inhalations. And third, they can build up indoors, where we actually spend most of our time. So, although we know airborne microplastics are important, our understanding is still quite limited. And the main reason for this, the way we measure them. Most existing sampling methods are not designed specifically for microplastics. They also collect many other airborne particles, which makes the analysis more complicated. So we often need extra preparation steps, and these can increase the risk of contamination or even particle loss. Another key issue is that there is no standardized method. So difficult studies reported their results in different ways, making it difficult to directly compare them, as we know. So this raises an important question: how can we sample airborne microplastics more effectively? To answer this, let's first look at the methods that are currently used. Traditionally, airborne microplastics have been collected using two main approaches. The first is passive deposition sampling, where particles simply settle on a surface over time. The second is active sampling, where air is pumped through a filter to capture airborne particles. However, these methods are originally designed for general particulate matter, not specifically for microplastics. And this difference becomes very important when we try to study airborne microplastics. So to understand why, we first need to think about how microplastics are different from typical airborne particles. Many microplastics appear as fibers or irregular fragments, and they don't behave in the same way as other particles in air. When traditional samplers are used, the filter collects a complex mixture, including dust, organic material, and other airborne particles. In this mixture, microplastics make up only a very small fraction, and this makes the analysis much more challenging. So, while these samplers work very well for particulate matter, they are not really designed for microplastics. And this is where our approach comes in. New allophilic active sampler. So the idea behind this sampler is actually quite simple. It's based on an oil-water separation principle. Instead of collecting particles on a filter, the system uses a mixture of water and oil. So microplastics are made of polymers, and these materials tend to interact more easily with oil than with water. So when air passes through the system, microplastic particles move into directly the oil layer and stay there. And this means that part of separation already happens during the samplings rather than later in laboratory. And this is one of the key advantages of this approach. So to test this idea in practice, we developed and evaluated the sampler under controlled conditions in the laboratory. We tested different oils and different sampler configuration to find the most suitable setup. We then carried out the recovery experiments using standard microplastics to see how efficiently the system could capture microplastics, airborne microplastics. And blank tests were also performed to check for any potential contamination during the sampling and processing. So here you can see that the sampler is able to recover different polymer types quite well. As you can see, the left-hand side graph, this is recovery efficiency, and at the same time, the blank results show very low background contamination because we already ignored the pre-treatment steps. We already separated microplastics from the particles during the sampling. So this gives us confidence that the sampler can capture microplastics reliably without introducing significant contamination. With this validation, we then move on to real indoor sampling. So for real sampling, we wanted to compare it with a commonly used approach in the airborne microplastics, which is the conventional deposition passive sampling. In deposition sampling, particles simply settle on a surface, as I mentioned before, over time. So what we measure here is what falls out of the air. But in contrast, our system actively draws air through the sampler, and microplastics are captured directly in the oil phase during the sampling. So instead of waiting for particles to settle, we are actively collecting what is actually in the air. To explore this difference, we apply both methods in the same indoor environments. For real sampling, we selected one of the most frequently used laboratories in our facility, where human activity is relatively high. The sampling campaign lasted 6 weeks, covering both working hours and non-working hours. This sampling period allows us to explore how human activity influences airborne microplastic levels. So both methods were running in parallel under the same indoor condition, and this gives us a direct comparison, efficiency comparison between the two approaches. After sampling, all samples were analyzed using pyrolysis gas chromatography mass spectrometry, or pyro-GCMS. Briefly, this technique allows us to identify and quantify different plastic polymers. We then look at which polymers were present and how their concentrations changed over time. So first, here we compare the total mass of polymers collected using the active sampler and passive deposition sampler. As you can see in the graph, both methods detected multiple polymer types across all sampling periods. However, the overall patterns are quite different between the two methods. And this is expected because, as I mentioned, the passive sampler collected settled particles, which is larger particles compared to suspended one. And while the active sampling captures what is actually in the air. We also see some variation between weeks, which likely reflects changes in indoor activity and conditions. So rather than giving the same information, these two methods provide complementary insights. And the other advantage to use the active sampling is that using the active sampler, we were able to calculate airborne microplastic concentration, mass concentration, since the sample air volume is known. This means we can express the results microgram per cubic meter. Here I'm focusing on the polymer composition based on these concentration measurements. Although the total concentration differ between working and non-working hours, the overall polymer profile remains quite similar. But interestingly, our field notes show that in some weeks, the lab was also used outside working hours. As we can check in the graph 4th and 5th week, we can see the results. They are really close to each other, working hour and non-working hours. So this is clearly reflected in the results. So this really highlights how sensitive the system is to changes in indoor activity. And it was exciting to see this so clearly in the data. We are now actually looking forward to applying this approach in different environments. So to better understand our results, we compared them with the literature in previous indoor studies. As you can see here, our results fall within the range reported in the literature, but at the same time, there is quite a wide variability across different environments. This is not surprising, as indoor microplastic levels are influenced by many factors such as human activity, ventilation, and also the type of the environment, the type of the space. So what we find particularly interesting is that even within a single indoor environment, we observe noticeable differences over time. And together with our field observations, this really highlights how dynamic the sampler is. For me, this is actually one of the most important exciting part of this research, being able to capture these small changes using this new approach. To conclude, there are still many unknowns in airborne microplastic research, and there is currently no fully standardized sampling method. With this in mind, in this study, we developed an active oleophilic sampling approach for airborne microplastics, and we showed that the system can effectively capture and quantify different polymer types. By applying the sampler in a real indoor environment, we were also able to explore how microplastic concentrations change over time and with human activity. And importantly, these results show that indoor microplastic levels are not consistent. They change depending on how we use and interact with our environments. This opens up new opportunities to better understand sources, exposure, and potential risk of airborne microplastics. And overall, I believe this work is a step towards improving how we monitor and understand airborne microplastics in both indoor and outdoor environments. So I would like to thank my supervisor, collaborators, funding bodies, and institutions for their support through this work. Thank you for your attention, and I would really happy to take any question. And if anyone is interested in this work or potential collaboration, please feel free to get in touch. I would like to be very happy. Thanks.
Thank you so much, Fatma, for showcasing your amazing research. So now we'll open the floor for any questions that the attendees may have. I think the first question goes to Lydia. It's from Michael Bramson. So Michael says, "Great talk, Lydia," which I agree. Michael is wondering what you meant by microfauna. Is it animals that can be seen by humans?
Well, that's not what I mean. That refers probably to this citation from the Scientific American. So I would have to go and read this, what they included, but I would imagine that they included, well, what we see, and this probably didn't include the cells and so on. But again, that's not really, I can comment without checking the reference.
Thanks, Lydia. Just to follow up on that question, Grace asked if there is a scope for animal studies in terms of airborne microplastics.
Well, there certainly is, but if so, it would have to be extremely well defined. The first question is, what is the purpose? Because there's been all the studies, reference we found. Everybody doing something different way, different methods, and responding to different questions. So if we say that if there's a range for studies, what questions do we want to answer? So for me, coming from air quality and from the risk of inhaled plastics, it is the question, what is the concentrations of plastics which are smaller than 10 micrometers. This must be measured by size distribution, because if we measure just by mass, we don't know what it is. So it must be size distribution. So this requires specific techniques and specific settings. again, if it is done like in a chamber environment with some artificially generated plastics, it may be completely irrelevant to real environments. So it would have to be done in a real environment with a technique measuring size distribution of the particles in the air, and with a capability to distinguish between which of these particles are plastics and which of these particles is something else, and understanding that the majority of the particles are not plastics. This is a huge, huge challenge, really.
Thank you so much, Lydia. For Fatma, there's a question from Nicholas. He's asking if you're using, did you use impactors before your oil sampler to remove larger particles?
Thank you for this great question, because actually at the moment I'm not using an impactor before the oily sampler. The system is designed to capture particles across a broad size range, including larger particles. However, using an impactor is something I'm considering for future work to better understand the size distribution, but at this moment I'm not using an impactor.
Thank you, Fatma. For Lydia, there's a question from Barnyam. They're asking, are the data from your review reproducible with the same sampling method, or the results varied significantly for the same sample?
Well, everybody doing this used a different method, and there was no comparison between the methods. So it would require a specifically designed studies to compare the methods. But we haven't found a study like this. So each study was a separate method reporting something else, and this wasn't compared.
Yeah, and just out of curiosity, did any of them use the oleophilic sampling method that Fatma presented?
I don't think so. I don't think so. Well, but this is all. The studies which we reviewed were with the specific purpose of particles smaller than 10 micrometers. So, so therefore, we looked at the methods which were capable of saying something about the size of the particles. So I'm not saying that there was nothing about this kind of methods, but we didn't look at the studies in which we couldn't find any information about the size of the particles. So that's an important point.
Thanks, Lydia. For Fatma, there's a question from Stephen. What proportion of indoor particulate pollution did the microplastics constitute? Did you measure the other indoor particulates at the same time?
Oh yeah, that's really good and deep question. Actually, the water oil system relies on the oleophilic behavior of most, influenced by properties such as hydrophobicity. But however, we do not explicitly quantify and control for this kind of properties, differences between the polymers. Regarding degradation, surface oxidation could increase hydrophilicity, which may reduce transfer to the oil phase. This is a limitation of the method, and it is something we are aware of. In our current results, we still detected a range of common polymers, suggesting that the system captures most particles effectively, but more degraded or hydrophilic particles may be underrepresented. Yeah.
Fatma, that's for Lydia. There's a question from Nicholas. He's saying that the health risk assessment will probably require a size distribution to be measured. How is this done for microplastics?
That's what I'm stressing, that size measurement of size is critical, because this is then related to inhalation. If the particles are too big to be inhaled, while we can find them present in indoor environment, like when we suspend dust, and we sometimes even see these particles, but if they are too big, we cannot inhale them. So they size distribution, to measure the size, to understand if they can be inhaled or not. That's critical. And then if we do this, but the problem here is that if we use the methods which can in sort of very high degree of detail tell us what the size distribution of particles in the air, like using scanning mobility particle sizer with well, size resolution of some nanometers, the point is that we see the size distribution of all particles in the air, but it is not telling us what fraction of these particles are plastics. So in addition to this size distribution measurements in that way, using this instrument, we would need another method, which would also be able to tell us what fraction are plastics. And that's where the complexity is.
Thank you, Lydia. That's a really great point. There's a question from Jeff. I guess this is a general question which either of you can answer. Regarding microplastics in the atmosphere, we are still some way from understanding how microplastics intersect with the environment and interact with human inhalation. How far are we from being able to make meaningful statements about the risk of microplastics for environmental systems and also for human health due to outdoor and/or indoor exposure?
I can start on this. We just 2 days ago, we had a little presentation in our group by another student who just looked and did some review, and that was the review was on what's in the brain in terms of microplastic, what was reported. That was for an and he found several studies reported on the concentration of plastics found in the brain. Huge differences, and some of them completely unrealistic. When we looked, I don't quite remember the percentage of this, so that looked like several percentage of the brain is plastic, which means that there's some problem with the methods of how it was done. So I haven't really seen something really meaningful and something which can be reproduced and agreed by some consensus of scientists looking from different angles of this, which would tell what in a quantitative way is the health risk.
Thank you, Lydia. Fatma, do you have any thoughts or do you agree with Lydia?
I totally agree with Lydia. I can say that we still in the early stages. We know microplastics are present in air and inhalation.
Sorry, Fatma, you're just breaking up a little bit.
Pardon. No, it's okay?
Yeah, that's fine.
Okay, I was talking just that. I totally agree with Lydia, and we are still in early stages. We know microplastics are present in the air and inhalation exposure occurs, but we lack standardized methods and sufficient data on exposure and also health effect. We were also working on the human matters, human exposure. So we are not yet able to make definite risk assessments, but the field is progressing quickly. Hopefully, we will figure out.
Thank you, Fatma. So I guess this will be the last question. It's a question, let me see. So for a question from Morawska, they're asking, did you digest the samples before analyzing them using PYGCMS?
Yeah, thank you for that. This question, Morawska. Actually, I did just we didn't digest the samples, but we have cleanup process because the oil can interference the system, this instrument system. So we need to remove the oily part on our filter. That's why I just used some chemicals, organic chemicals, to remove the oils, but at the same time to protect the microplastics. Yeah. Then we just run the samples directly in this PyroGCMS.
Okay, thanks, Fatma, for
Yeah.
answering that last question. So I think we can conclude today's webinar. Before I do, I'd just like to extend my sincere appreciation to Lydia and Fatma for sharing their time and insights on this topic. We hope our attendees have found the discussion valuable and informative. If you have any further questions or would like additional resources, please don't hesitate to reach out to the Center for Safe Air. We look forward to seeing you at our next webinar. Thank you and have a great rest of the week. Thank you so much, everyone.
Thank you very much.
Bye.
Sabrina. Thank you, Lydia.
Bye.